Welcome to part 2 of your lecture on learning and memory. In this part of the lecture, we're going to focus on relational learning and long-term potentiation. Now we're going to consider the role of the cortex in perceptual learning. Here's that same handy diagram again. So information from the thalamus is sent to the extrastriate cortex and then to the visual association cortex. The visual association cortex is divided into two streams. There's a ventral stream and a dorsal stream. The ventral stream is involved with object recognition, so basically the what that you're seeing. The dorsal stream is involved with perception of the location of objects, so where they are. As you might imagine, one of the most important things that we can learn, particularly in early life, is to recognize our kin and our primary caregivers more specifically. So perceptual learning is also a part of this, and the cortex plays an important role here. In terms of memory, perceptual learning causes changes in the neural circuits of memory. These become active again when the same stimulus is presented. It constitutes the recognition of this stimulus or the readout of the visual memory. So we're able to imagine familiar faces, for instance. Specific kinds of visual information can activate certain regions of the extrastriate cortex. And retrieval of memories in the form of pictures and sounds and movements or spatial locations have the effect of activating the appropriate regions of the sensory association cortex. This figure from your textbook posits the location of these where and what pathways. And you can see here that stimulation of neural activity in the extrastriate by visual stimuli seen at a later time is shown. The eye sees the stimulus. The activity reaches the primary visual pathway ventral to the thalamus. The what of the stimulus is shown in the ventral stream, while the where of the stimulus is shown in the dorsal stream. In any event, activation of neural circuits in the sensory association cortex constitutes the readout of perceptual memory. In this illustration, you can see evidence of retrieval of visual memories in participants who looked at photographs of static scenes or scenes that implied motion, like what is shown here. So the x-axis shows 3 types of motion. The y-axis shows percentage of change in signal, which represents the level of activation as measured by fMRI, MT, or MST. In all these cases, a region of the visual association cortex corresponding to movement is implied. And now we're going to discuss 2 aspects of relational learning. One, human anterior grade amnesia, which we'll explore using a couple of different case studies in class, and also relational learning in laboratory animals. Allow me to draw your attention to some important multimedia on the Pearson site. One detailed simulation of the case of Clive Waring, and another on how memories are formed. There are 2 kinds of amnesia. There's retrograde amnesia and anterograde amnesia. Retro has to do with anything that happened before the brain damage, and anti has to do with anything that comes after the brain damage. This infographic depicts the learning process, and in particular, memory consolidation. So first we have sensory information. It enters the short-term memory. Rehearsal keeps it there, and then the information makes its way into long-term memory where it is permanently stored. The conversion of short-term memories into long-term memories is labeled consolidation. There are a couple of ways to think about consolidation in order to really conceptualize this. It's good to think about the two stages of memory. So short-term memory involving storing of limited amounts of information temporarily, long-term memory storing of unlimited amounts of information more permanently. Consolidation capitalizes on hippocampal functions, particularly the hippocampal formation. Some spared learning abilities are notable. In patients with anterograde amnesia, we see that they are capable of 3 of the 4 types of learning. So they can perform perceptual learning, stimulus response learning, and motor learning. Studies also show evidence of long-term retention. As you know, based on the complexity of human behavior, most of what we do involves multiple forms of learning. In this way, amnesia is a really curious phenomenon because although people with amnesia can learn to perform tasks, they don't always remember having learned them. And people with amnesia, particularly anterograde amnesia, are not able to form declarative memories, and yet they can recall non-declarative memories. With the cerebral cortex, the hippocampus is very important for long-term memory storage. And in this case, the hippocampal formation is especially important for the consolidation of declarative memories. The hippocampus receives information about what is going on from both sensory and motor association cortices. It processes this information, and then through its efferent within E connections with these regions, it actually changes the memories that are being consolidated, linking them together so that we can remember the relationships among elements of the memories. In this illustration from your book, you can see that in a particular fMRI study, the retrieval of the youngest long-term memories activated the hippocampus more than the cortex of the superior frontal gyrus, and that contrastingly, the retrieval of older memories activated the hippocampus less than the cortex until about 9 years, where both regions were activated. So it's interesting to note that this unfolds in a time-dependent fashion, but that younger memories and older memories are somewhat divergent in terms of their hippocampal activation. In contrast to episodic memory, which has to do with specific events that you've experienced, semantic memories are a type of declarative memory that can be gradually acquired over time. It involves facts, but not necessarily information about the context in which the facts were learned, and it's stored in the anterolateral temporal lobe of the neocortex. Spatial memory involves functions of the right hippocampal formation, as well as the caudate nucleus. The right hippocampal formation becomes active when a person remembers or performs, for instance, a navigational tasks. So think of those London cabbies, the black cab drivers that we talked about. People with damage can still remember some consolidated memories, but the hippocampus is required for detailed levels of spatial navigation, obviously in keeping with the discovery of its enlargement with increased experiences navigating metropolitan London. The hippocampus is used for spatial strategy, so it makes sense that it would increase with years of study in terms of acquiring the knowledge for black cab drivers in London. And the caudate nucleus is specifically involved in the response strategy. Some of our understanding about the different roles of the caudate nucleus and the hippocampus with regard to spatial versus response strategies in navigation comes from a 2007 study that looked at basically errors in navigation in relation to gray matter and showed that with increased gray matter in the caudate nucleus, you get fewer errors, while with increased gray matter in the hippocampus, you get more errors, specifically when it comes to measures of spatial versus response strategies. In your textbook is highlighted the case of Henry Gustave Molaison, who lived from February 26, 1926 to December 2nd, 2008, and was known widely as HM. And HM is probably the best known single patient, well, besides Phineas Gage, in the history of neuroscience. His severe memory impairment, as you may recall, resulted from an experimental neurosurgery procedure to control his seizures that emerged after a bicycle accident. He was studied subsequently for 5 decades and really is somewhat of a medical marvel considering his bilateral medial temporal lobectomy. This image from your text compares the temporal lobe and the hippocampus of a typical brain with those of HM. A cross-section along the temporal lobe of the typical brain is shown on the left. At the right, we have a bilateral medial temporal lobe lesion, which can be seen in HM's brain. A typical hippocampus is shown on the left, and on the right, HM's hippocampus, which shows lesions along the region. Spatial learning has been widely studied in laboratory animals, and these show problems in navigation, specifically following hippocampal lesioning, as you might expect. With different neurons having different spatial receptive fields, these animals respond differently depending on the location of the lesioning, the injury to the hippocampus. The neurons specifically are called place cells, and information is represented by particular patterns of activity in circuits of large numbers of neurons. The hippocampus, when it comes to spatial learning, receives information from the parietal lobes by means of the entorhinal cortex. This contains grid cells, head direction cells, and border cells. Information provided by these cells reflects the animal's location as well as their head's direction. And activity in the neurons encodes the current location as well as the intended destination. In this 3-part illustration, you can see that the firing rate of a border cell in the rectangular chamber and in the same chamber that was elongated in a horizontal or vertical direction varies. In all 3 parts, the rectangular areas show the hottest colors along the right walls. So this is what activity of a hippocampal border cell looks like. Hippocampal place cells encode more than just spatial information. As you can see from this illustration, which shows the T-maze alternate trails, there's a call out pointing to a dot at one end of the trail, which reads animal starts here, and there's a call out pointing to a corridor connecting the goal boxes that read stem of T-maze. The left turn trial starts at the bottom and ends at the top of the other end. Similarly, the right turn trial starts at the top and ends at the bottom on the other end. Rats are trained essentially to turn right and to turn left at the end of the stem of the T maze on alternate trials, and their paths are shown. And now we're going to turn to the induction of long-term potentiation, focusing specifically on the role of NMDA receptors, AMPA receptors, and also synaptic changes. First, I want to draw your attention to the number of really helpful simulations that are available on the Pearson website that illustrate these concepts for you. Long-term potentiation is the cellular basis of learning and memory. It increases in excitability of a neuron to particular synaptic input caused by repeating high-frequency activity of that input. The primary input to the hippocampal formation comes from the entorhinal cortex, which is shown at the base of this cross-section that's shaped like a human heart. The perforant pathway depolarizes either by exposure to stimuli in the environment or by delivery of a burst of pulses. One of the most clear and succinct descriptions or definitions of long-term potentiation that I've seen actually comes from Wikipedia, which states that in neuroscience, long-term potentiation is a persistent strengthening of synapses based on recent patterns of activity. These patterns of activity, synaptic activity that is, produce a long-lasting increase in signal transmission between two neurons. And that essentially is what you see depicted in the bottom illustration here, where before and after long-term potentiation, we see a big difference, but there is very little difference to be seen after long-term potentiation when you go from 1 hour to 24, 48, 72, and 96 hours. Neurons that are wired together in support of a memory fire together. Basically, long-term potentiation, or LTP, happens when presynaptic and postsynaptic cells are depolarized at the same time. As you will recall from your reading, the NMDA receptor, which is a neurotransmitter and voltage dependent ion channel, is critically important for learning and memory by virtue of its involvement in the mediation of magnesium and calcium levels inside and outside the cell, the hippocampal neuron, for instance. So with the NMDA receptor, if a molecule of glutamate binds to it, the calcium channel cannot open because the magnesium blocks the channel. Depolarization of the membrane boots magnesium away and unblocks the channel. This allows glutamate to open the ion channel and permit the entry of calcium ions, again as part of depolarization. It's important to note that there is an associative component of long-term potentiation. This means that if the activity of strong synapses is enough to trigger an action potential in the neuron, the dendritic spike will depolarize the membrane of the dendritic spines, priming NMDA receptors so that any weak synapses active at the time will become strengthened. So, the company that neurons keep, whether or not they are wired together, dictates the extent to which memory or some piece of knowledge can become embedded. So the activity of strong synapses cause the cells to fire, and a dendritic spike depolarizes the postsynaptic membrane, causing this associative long-term potentiation illustrated by this image. Synapses are strengthened. And this has to do with certain conditions for long-term potentiation being met, particularly calcium ions entering the dendritic spine through the NMDA receptor through the process just explained. The calcium ions activate enzymes in the spine, and these activated enzymes cause AMPA receptors to move into the spine. Finally, an increased number of AMPA receptors in the postsynaptic membrane strengthens the synapse. In keeping with the idea that synaptic changes happen over an extended period, LTP changes the size and shape of dendritic spines and can even cause the growth of new dendritic spines. So in this image, you can see that at half an hour before electrical stimulation, there's no growth of the dendritic spine. Then at 2 hours after electrical stimulation, and then 21 hours after electrical stimulation, we can see a sort of mushrooming of the dendritic spines. The opposite of long-term potentiation is long-term depression, and this has to do with the weakening of synapses that are no longer useful to the human or animal. So with long-term depression, we have essentially an activity-dependent reduction in the efficacy of neuronal synapses, and this is mediated by basically the same mechanisms, NMDA and AMPA, or AMPA as some people pronounce it. So with LTD, however, there's not enough depolarization to cause widespread removal of magnesium. There is some removal of magnesium, and the low level of calcium that is allowed to enter the cell and lead to cellular changes actually instead leads to some synaptic weakening through mechanisms that are still being defined, but in part involve the removal of glutamate receptors. And so I have this illustration here to remind you that there's a really good 2-minute neuroscience video describing this interesting phenomenon.